always inorganic. And 10,000 Dalton. So what is the one more thing? Uh, the inorganic molecule, their mass is 10,000 Dalton and above, right? And they are larger. Fine. Then we come to organic part, the organic biomolecule. When we talk about the organic biomolecule on the basis of their mass, we divide them into two categories, or rather than size, we can say on the basis of their size, we divide them in two categories, right? Macromolecule and micromolecule. Macromolecule, for sake of example, DNA, and micromolecule, they're smaller in size, right? When we talk about the smaller in size, so their weight is, or their size is 18 to 800 Dalton, right? For sake of example, sugar, amino acids. So today we will discuss about the micromolecules, right? We'll discuss about the carbohydrate means sugar. We'll discuss about the protein means amino acid, right? We'll discuss about the fat means lipid molecule, right? So, and we will also discuss about the DNA as well, right? So first we will discuss about these micromolecule, then macromolecule. So organic molecule, when we talk about the organic molecule, what are the example of organic molecule? Or rather than example, what are the organic molecule which basically, major organic molecule which basically found in cell, right? Protein, carbohydrate, fat, lipid. When we talk about the nucleic acid, the full form of DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So at the end of both, there is a suffix nucleic acid. So collectively, we call them nucleic acid. So when you get a term collectively nucleic acid, it means we are talking about the DNA and RNA collectively, right? And last but not the least, enzyme. So in our syllabus today, what we will discuss, that is a protein, carbohydrate, fat or lipid, nucleic acid and enzyme, right? So first, please note down what are the biomolecules. Copy paste as it is written over there, right? Then I will dictate some. The heading will be biochemistry option, right? Let me know when you note it down. Done? Martin? Alia, done?
Yes, sir.
Okay, great. Martin, done?
Yeah.
Great. So now please note it down. Put a sentence below this one. Chemicals or molecule present in the living organism present in the living organism are known as biomolecule. Full stop. The collection or sum total of different types of biomolecules compound and ion present in the cell is called cellular pool. Pool. Full stop. Next point. Biomolecules can be of two types. Biomolecules can be of two types: inorganic and organic. Biomolecule can be of two types: inorganic and organic. Now the inorganic and organic type you have already gone through with that, right? You have already noted it down, right? So no need to note down. And then next point. Depending on molecular weight and solubility. Depending on molecular weight and solubility, organic molecule fall into two categories: micromolecule and macromolecule. Next point. Microbiomolecules, microbiomolecules are small sized simple chemical that have low molecular weight in between 18 to 800 Dalton. In between 18 to 800 Dalton. That is less than 1000 Dalton. Next, macromolecule or macromolecule, macro, MACRO. Bio macromolecule are large in size, with high molecular weight, full stop. Usually their weight is 10,000 Dalton. Usually their weight is 10,000 Dalton and above. 10,000 Dalton and above, except lipid. So that is basically the question of medical examination. So lipid is macromolecule. They ask you what is the lipid. Lipid is macromolecule, but their weight is less than 10,000 Dalton. Still, they are considered as macromolecule. So you have to remember that lipid is macromolecule, right? These are found in acid insoluble. These are found in acid insoluble fraction and acid insoluble fraction, and they are always organic. Right. So this was about the macromolecule, micromolecule, and what are the biomolecule. Clear? So the next thing, that is the average composition of living cell. So when I say what is the average composition of living cell, so look, average composition of living cell, the content water is 70 to 90%. So if we talk about the chemical composition of cell, water makes 70 to 90% part of the cell. Protein makes 10 to 15%, right? Carbohydrate 3%, lipid 2%, nucleic acid 5 to 7%, and ions 1%. Please note down this composition. Please note down quickly. Note down quickly and then let me know. But the most important question, which we will discuss in next PPT, that is what are the secondary metabolites. So normally you will get the question as a 2-marker question in your school examination. What are the secondary metabolites? And give their example. That kind of a question. Done. Note it down. Alia, done? Martin?
Yes, sir.
Okay. Again, both of you, tomorrow is Friday. Martin, this time I checked your copy. There was nothing to check over there. Nothing to check. So, first thing, write down your name, date of test, number of test is second, first, what the test is. Next, the answer number. Next, there should be proper answer and proper scanning. Scan it properly, just don't click the photo and send it. Don't do that. Got it? Martin,
Yes.
give the test properly this time. I'll check.
Properly as a
Look, let me. I will check you in front of you only. Look, that's how you write. Look, that's how you scan. Martin, what is there
or
to
I
check
should
this?
scan properly. Yeah,
Yeah, no. Look. This is the section B means 2-marker question. You have just written two words. So do you think that
Oh,
I will
I
give
did
you?
not know the answer.
So don't do that. Look, scan it properly. Look, question number 10. You have written 2 lines, then you have cut down, then this is. I do not expect this kind of the answer from a student like you. I expect better from your end. Look, section D, nothing is there. I considered you good student, so I have much more expectation than you. Alia, always appear in the test. Do you
Yes,
join
sir.
the test? Have you
Yes,
joined the
sir.
test? I haven't
Yes,
got your look. I haven't your copy, single copy in these 3 tests.
sir. I gave first two tests. The last one I didn't because I was
Okay.
attending some function. So
So this Friday on tomorrow there is a
yeah,
test,
yeah.
right? I expect
Yes, sir.
your copy, right, Alia? I'll
Yes,
ask
sir.
you. Fine.
Yes,
Martin,
yes.
hope you will revise the concept and give it properly.
Yes,
Got it? And tomorrow test, that is the simple one. Tomorrow test, that is a simpler one. Because tomorrow I'll ask you this. I have given you syllabus for the cell, the unit of life, right? That is the syllabus for tomorrow's test, right? And even the questions are not that much difficult. When the questions are very easy, right? So okay,
yeah,
fine. So first, what are the metabolite, right? So there are 2 types of the metabolite: primary metabolite and secondary metabolite, right? So metabolites they are basically organic compound, fine, which takes part in reaction or metabolic reaction. They are called organic. And primary metabolite are basically those metabolites which directly take part in our reaction, right? Chemical reaction, or you can say organic reaction, right? Or cellular reaction. And secondary metabolites are those which do not directly participate in any metabolic reaction. They are called secondary metabolites. Sometimes they have different purpose. So yes, if secondary metabolites are absent in the cell will survive. But without primary metabolites, cell cannot survive, because primary metabolites are those compounds, those chemicals, those organic compounds which directly participate in reaction. For sake of example, if we talk about the plant, right? So primary metabolites are those which directly involve in the photosynthesis, which directly involve in the respiration. We talk about the humans. Primary metabolites are those which directly involve in the digestion process, directly involved in the respiratory process, right? So they are the primary metabolites. So without primary metabolite, any organism cannot survive, either it's plant or animal, right? Clear? So put the heading. First heading is metabolite. Uh, the organic compound, like carbohydrate, fat, protein, nucleic acid, etc., are required for their basic basic metabolic processes. and are found and are found throughout the and are found throughout the plant kingdom. Throughout the plant kingdom are called primary metabolite. Full stop. Next, this is important. What are the metabolites or primary metabolite and secondary metabolite, right? For your school exams, and you can put the example. This is a 2-marker question. Next, the organic compound. The organic compound which are not directly involved in which are not directly involved in primary metabolism like photosynthesis, respiration, etc. and seems to have no direct function and seems to have no direct function in growth and development of plant. Growth and development of plant. Such compound are called secondary metabolite. Example, you will get question, what are the secondary metabolite? Give the example. Pigment, which gives color and meant for pollination. Alkaloids, they may be defensive or they gives particular peculiarity to particular plant. Essential oil, toxins, drugs. Note down the example. Secondary metabolite. Done? Noted? Done, Martin?
Yeah, yes.
So come to the first this one biomolecule, that is protein in RNCRT. First, I will make you understand that what the protein is. Everything is made up of a small unit, and we call them monomer. You must be sitting in a room, and that room is made up of brick. So that room can be called a particular compound, and every brick is called monomer. In the same way, protein are made up of some monomer. So the basic unit of protein, try to understand, and you have to note down each and everything like this way. Look, basic unit of protein is amino acid. Look. So we can say monomer of protein is amino acid. So basically, every protein is made up of small molecules, and those small molecule or basic unit are amino acid. 1, 4, 5, 6, 7, like this kind of. One amino acid combined with another amino acid, right? the bond between two amino acids are known as peptide bond, right? So we can say that protein molecules or protein monomer remains connected with each other with the help of peptide bond, right? And they make a long chain. When amino acid or when monomer of protein, they make a long chain. This long chain is called polypeptide chain. Clear? Now this polypeptide chain, what happened? Folding of polypeptide chain takes place. When polypeptide chain fold, right? So this folding is folded polypeptide chain is called protein, right? And this folding is supported by these kind of structure. They are called disulfide bridge. So with the help of disulfide bridge, this remain interconnected, right? This protein. And the folding of protein will decide or folding of protein decide the nature of any protein, what exactly this protein is going to do. Clear? Protein may be structural protein, means that makes the structure of body. Protein may be enzyme, protein may be hormone. So the nature, what the protein will do, that will be decided by the folding of protein. Did you got it? Tell me. Keep on telling me. Got it or not? Martin, clear? So please draw it. Monomer, peptide bond, right? Peptide bond, polypeptide chain, and protein. Till this point, first draw it. Then I will tell you what is the peptide bond, how the peptide bond formation takes place. That is important. Note down quickly. Let me know when you note it down. Done? Martin, note it down. Alia,
Yes.
Alia done? Alia, are you there?
Yes, sir. Yes, sir. Done.
Okay, fine. So now I want your 100% focus over here. Look, try to understand what is the structure of amino acid. So this is the one amino acid molecule. This is the structure. So that is a carbon compound. This is amide group, NH2. HNH, right? So in short, we can say this is NH2. This is hydrogen, right? And this is carboxylic group, right? COOH, right? Here you can say this R. R is an alkyl group. Alkyl group is a hydrocarbon chain. It may be of two carbon compound, 3, 4, 5, 6, any length, right? So that depends on compound to compound or amino acid to amino acid. So the R is alkyl group. This is amide group. This is carboxylic group, right? And this is hydrogen. Here what happened? This is another amino acid. Two amino acid come closer to each other, right? OH means hydroxyl group from here and hydrogen from here is removed. H and OH combine, they make H2O. So water is removed, means this is dehydration, right? This dehydration takes place, right? Water molecule is removed. Now there will be a bond between them, right? So again, and H and H, this amide group, alkyl group, hydrogen. Here the bond, this will be carbon, CO and NH. So this is CONH, they will make a bond. This is called peptide bond, right? Again, this is alkyl group, this is carboxylic group, this is the hydrogen. So the bond CONH, this bond is called peptide bond. That's how the peptide bond takes place. And when the peptide bond formation takes place, hydrolysis will take place. Means the removal of water molecule will take place. So whenever one peptide bond will be formed, one water molecule will be removed. Is that clear? Did you got it? How does the peptide bond formation takes place?
Yes, sir.
Please note down as it is. And put a heading. Peptide bond formation. Peptide bond formation between two amino acid molecule.
So OH and H is H2O.
Yes, yeah. OH and H, they will combine, they will make H2O molecule. And H2O molecule will be removed during this reaction. Even here in this reaction, here you can write it down H2O plus Noted? Done? Alia? Martin?
Yes, sir.
Okay, great. So, understood how the peptide bond formation takes place? Both of you?
Yes, sir.
Great. So now start writing down, right? Not down some point. So basically, they are micromolecule. Means proteins are micromolecule. They may have one or more than one polypeptide chain. So one protein can have one polypeptide chain or more than one polypeptide chain, right? A single polypeptide chain. Suppose that there is a protein, it have single polypeptide chain. So a single polypeptide chain can be 50 amino acid larger, right? Not less than that. Okay. They are the most abundant macromolecule, right? Out of all the macromolecule, protein is most abundant. 50% weight of the cell is made up of proteins, right? Collagen fiber, that is the most abundant protein in animal. So can you tell me what is the collagen fiber? Any of you? What is collagen fiber? Alia, any idea what is collagen fiber? Martin? Have any idea?
Yeah, what, sir?
What is collagen fiber? What are the collagen fiber?
It's a protein.
Yeah, that's protein. It is written over there. Any example? Can you give me the practical example of collagen fiber? You know,
Amino acid.
your leather bag, the leather which you call leather, that is made up of
Leather.
collagen fiber. Leather. You must have leather purse, leather wallet. Yeah. You know, the leather, that is the animal leather. That is basically made up of collagen fibers. And the more collagen fiber in the leather, that leather will be more stronger, right? So collagen fibers are basically found in the skin, right? So skin of animal, skin of human, in our skin, that's collagen fibers are found, right? Collagen fibers give the stretchability and strength to skin, right? Buffalo, cow, they have thick skin because they have the lots of collagen fiber are over there, right? So collagen fiber decide the strength of any leather, right? So what is the leather? Leather is basically made up of collagen fiber, and collagen is a type of animal protein. Fine. So collagen is most abundant protein in animal. The most abundant protein which is found in animal, that is a collagen. Rubisco. This question I've been found number of the time in medical examination. Rubisco actually ribulose biphosphate carboxylase oxygenase. This is the most abundant enzyme or protein found on the earth. This is a plant enzyme, right? Rubisco is the most abundant protein and most abundant enzyme found in the on the earth, right? So these were some fact about the protein. Start writing now. Protein are large sized. Protein are large sized macromolecules. Large sized macromolecule having one or more polypeptide chain, one or more polypeptide chain of amino acid. Full stop. The term polypeptide is often the term polypeptide is often used interchangeably. The term polypeptide often used interchangeably for protein. Both are same thing. Either you called it polypeptide or you called it protein. Both are same. So that is interchangeable word, right? Protein and polypeptide. Hope it must be clear. Next point. A single polypeptide. So what can be the length of a polypeptide? Minimum length of a polypeptide. A single polypeptide must be a single polypeptide must be at least 50 amino acid long, 50 amino acid long, in order to qualify this term. In order to qualify this term. Next point. Proteins are the most abundant. Proteins are the most abundant and most varied. Protein are the most abundant and most varied macromolecule which constitute macromolecule which constitute approximately 50%, which constitute approximately 50% dry weight of cell. 50% dry weight of cell. Clear? Next point. We are very close to chimpanzees. So the genes are chimpanzees almost similar to the genes of human. In the same way, we are, because chimpanzee and we are very close to each other. So the protein of chimpanzee will also be similar to the protein of human, right? So please note down. Closely related species may have similar protein. Closely related species may have similar protein, while unrelated species may have different protein. Right? So this was just a minute. Protein. I think that should be the structure of protein. Where is that? Okay. So you can note it down. The collagen is the most abundant protein, note down, and rubisco is the most abundant protein on the earth. Note down these two things. Then I will tell you the type of protein. Done? Look, there may be primary protein, secondary protein, and tertiary protein, right? Try to understand. If there is a, so then you have to put the heading types of protein. Look, primary protein, there is a single chain, this helical chain, and the length of a whorl may be 0.3 mm, right? So, not mm, nm, right? Nanometers. This is nm, nanometer, right? So, again, the secondary, so this is primary protein. There will be single chain, right? Now, in secondary protein, you can see they may be of two types. So, when chain start folded down, right? So, this is 3D structure. So, secondary protein may be made up of more than one chain, right? And this may be in the form of alpha helix, or there may be beta plated. In alpha helix, what happened? There is this kind of three-dimensional whorl, spring-like structure, but between these whorl, right? Between this whorl, there will be a bond. This is disulfide bridge, right? They are made up of disulfide bridge or hydrogen bonded, right? Then this there are, you can see there are three polypeptide chain. This black color structure is every polypeptide chain. These polypeptide chain are present in the form of sheets, and these all chains are interconnected with each other with the help of hydrogen bond, right? So they make beta plated. So secondary structure may either be alpha helix or maybe beta plated sheet, right? Then there is a tertiary structure. So in tertiary structure, this is the structure. There are hydrogen bond. There are some ionic bond between the polypeptide chain, right? And there will be disulfide bridge, right? So this is the tertiary structure. Fine. And what happened? This when you use egg, so egg is basically liquid form. So why it is liquid form? Basically the egg protein that is in tertiary structure, that is in liquid form. When you put in the pan, it becomes solid. Why it becomes solid? Because that tertiary structure, due to the heat, sulfur bonds start breaking down. That's why the smell come from the smell which come from the egg, that is of sulfur, right? The disulfide bond start breaking down, and that tertiary protein turn into primary protein, right? So that liquid egg is, that is a tertiary protein. That omelette is, that is the primary structure of protein. That's why it becomes solid. And then quaternary structure, hemoglobin is quaternary structure. It is made up of 4 type of polypeptide chain, right? 2 alpha chain and 2 beta chain. Clear? Did you got it or not? That is the simple. We have not to go much deeper than that. So this is sufficient at your level. Tell me, is it clear or not? Structure of protein, primary, secondary, and tertiary structure. Alia, Martin, tell me, please.
Yes, sir.
Okay, so start writing down. Put the heading, types of protein. First, primary structure, and draw the diagram. One more thing. At this end, means the left side, there will always be nitrogen at the end, and there will be carbon. Note down. In primary protein, the number and sequence of amino acid is decided by DNA. Full stop. The distance between two adjacent polypeptide bond is about 0.35 nanometer. Right? Next, secondary structure. Amino acid, secondary structure. Amino acid are present in linear sequence inside the polypeptide. Full stop. There are three type of secondary structure: alpha, alpha helix, beta sheet, and collagen. Alpha helix, beta sheet, and collagen. Full stop. In alpha helix, next point. In alpha helix, polypeptide chain. In alpha helix, polypeptide chain coiled spirally. Generally, in right hand manner. Full stop. Next point. In beta plated secondary structure, two or more polypeptide chain get interconnected by hydrogen bond. Full stop. A sheet is produced instead of fiber like alpha helix. Example, silk. So silk is example of silk. Beta carotene. That is example, right? Next, in collagen, polypeptide coiled around one another. In collagen, polypeptide coiled around one another. Full stop. The coil is strengthened by hydrogen bond. Next heading, tertiary structure. Draw both diagram. Secondary structure, draw alpha helix and beta sheet. Please draw it. Draw both of the diagram. Whenever types of the protein are asked, if you are writing the answer, then you have to draw all these diagrams. Irrespective of question ask you or not. Let me know when you draw it. Done? Alia, Martin, noted this structure? Hello. Alia, noted this structure or not?
Yes, sir.
Okay, Martin, what about you?
Yes.
Okay, so come to the tertiary structure. There is a bending and folding of, there is a bending and folding of various type of, there is bending and folding of various type of, various type of sphere, rod or fiber. Sphere, rod or fiber. Full stop. Tertiary structure is stabilized by, tertiary structure is stabilized by several type of bond. Tertiary structure is stabilized by various type of bond, like hydrogen bond, comma, ionic bond, comma, van der Waal, comma, covalent bond, full stop. Example, write down example. Myoglobin protein. Example, myoglobin protein. They are found in our muscles. They are like hemoglobin. They are found in our muscles, myoglobin. Right. Last but not the least, quaternary structure. Quaternary structure. First, draw that this diagram, tertiary structure. Draw this diagram quickly. Don't take too much time. Quaternary structure also write down the example of hemoglobin. Done. Martin. Okay, done. I didn't listen you. Put the heading quaternary structure. This is found only in, this is found quaternary structure. This is found only in multimeric multimeric protein. multimeric protein. Full stop. The different subunit can different subunit can fit together to give conformation. Example is hemoglobin, right? So this was all about types of protein. Go through with it and ask me. And if you have any doubt, in next class I will start with. Today we have done till protein, and secondly primary metabolite we did, and protein. Next class we'll start from the polysaccharide on, right? Ask me if you have any doubt. Go through with it. Go through with your NCERT and ask me if you have any doubt.
So what's of the different?
What different subunit?
Okay. Okay, thank you.
Welcome. Go through with it and ask me if you have doubt. Next class we'll start from the carbohydrate onward, right? Ask me doubt, then we will wind up.